I want to introduce our first keynote speaker, who I'm thrilled is joining us. Ananda Goldrath is a professor in the molecular biology section and a Tata Chancellor's professor in the Division of Biological Sciences at the University of California, San Diego, where she joined the faculty in 2004. In 2011, Ananda was named a Leukemia and Lymphoma Society Scholar, and in 2007, she was named a Pew Scholar. She's also a recipient of the Cancer Research Institute New Investigator Award, the V Scholar Award, the Julia Brown Faculty Fellowship, and the NIH Transformative Research Award. Ananda has really made critical contributions for us, for our understanding of transcriptional networks that control the formation, maintenance, and functional capacity of memory T cells. And her lab has identified transcription factors that control memory versus effector T cell differentiation. And I'll let her tell you more, but I am excited, when we're thinking about this new field of genomic immunology, Ananda is clearly one of the people who has helped to define it. And I'm glad that she's here to help kick off this event today. So I'll hand this over to Ananda.
Wonderful. It's so wonderful to be here. And Alex, I love the vision for this new institute, and sign me up. It really expresses so well how we've organized ourselves to approach science over the last 15 years, and you've put it all in one place. And I really envy the group of people that are participating, and I'm glad to be a part of it today. All right, I'm going to get my screen up here. And so I'm excited to tell you about how we've really been cellular immunologists, and as Alex said, a very traditional cellular immunology lab that has been benefited from exploiting genomic approaches. I'm sorry, you never know exactly what's going to happen, and my video popped into a completely different location, so get myself settled. All right, so I did note, however, that as I kind of represent the old guard, so the rest of the day is really the future of genomic immunology, and I love that it's highlighting, this symposium is highlighting the trendsetters and the people who are really taking us to realize the full potential of these phenomenal techniques. But that does make me a little bit the old lady, and so I'll try and set the stage so that we can really see where it's going over the course of the rest of the day. And so today I'm going to tell you a series of stories associated with our interest in understanding tissue residence and how cells accommodate adaptations to unique tissues. And largely the immune system has been studied in the past in the context of the blood and the spleen. And it's become quite clear that we need to understand the specific signals and context in which immune cells do their job. And in this case, we'll be focusing on CD8 T cells. And I'll tell you a bit about our efforts to understand the transcriptional programs that regulate their differentiation in the context of both infection and tumors. I'll highlight the work of the wonderful people who have done this along the way, as well as our collaborators. And I will disclose that I am on the SAB of Pandion Therapeutics and Arsenal Bio. All right, so we've really, over the years, used CD8 T cells as a model to study CD8 cell fate decisions. They're extremely accessible. They really are just a wonderful model system for understanding how a single cell, in this case a naive T cell, can receive a variety of signals, integrate those in the nucleus in the context of transcriptional programs that drive not only phenomenal differentiation and acquisition of effector functions, which we know are essential for clearing infection and tumors, but also to differentiate into a range of long-lived cell fates that provide unique functional activities that are essential for host protection. And so we know that, as I said, based on a range of factors, intrinsic and extrinsic contexts, a single naive T cell can give rise to a range of fates, a population of cells that will die after the infection is cleared, other long-lived populations that can give rise to secondary memory. And also these cells then seed a memory compartment in a range of tissues that provide protection really at the barrier sites where reinfection can occur. And so largely the field for a long time had studied those cells that are easy to access. And those heterogeneous populations that I pointed out are found within the spleen and the blood. And more recently, it's been appreciated that there's really the majority of memory CD8 T cells are lodged within tissues. And these tissue resident memory cells are really the majority of cells that make up a secondary response within the tissue, and they can serve a variety of functions to bring in circulating cells. But it became clear that we really needed to take a step back in how we've been looking at T cell memory and consider those populations. And to emphasize that point, I wanted to highlight some beautiful studies by David Mathis' lab, and this has been shown by a number of labs in a variety of contexts for both human as well as mouse. But what's clear is that there's a recirculating population of memory cells which can normalize between an immune host and a naive host. So in parabiosis studies, we can see that memory T cells within the spleen and lymph node normalize very quickly between the immune and the naive host. However, if you look at the tissue resident memory cells, so the memory T cells that are lodged within a whole variety of organs after infection and provide that barrier protection, this is not the case. There's not recirculation of these memory populations. And David's lab did a beautiful job enumerating this across a whole range of tissues. And so this graph shows an enormous amount of work, which showed that really between 85 and 90% in most, and 100% in most tissues of T cells that are in those tissues are in fact stuck there, and they're not recirculating, and we don't sample them and we don't study them when we look in the spleen and the blood. And so at that point, we decided we needed to take a step back in how we'd been approaching immune memory and to look at these cells more carefully. So these cells, as I said, provide early sentinel protection at sites of reinfection. They can protect against both pathogen as well as tumor growth. And so understanding how to lodge these cells in the right places is a huge potential for enhancing immunity and immunotherapies. These cells can immediately kill infected cells, as well as recruit innate and adaptive cells to the site of infection to amplify a secondary immune response. Defining what a memory T cell, a tissue resident memory T cell, has been challenging. Largely, the definition that I prefer are cells that don't recirculate, but not every experiment can be done with parabiosis. And so most of these cells express CD69 and many express CD103. What's clear is that T cells that differentiate into tissue resident memory cells need to downregulate KLF2 and thus its target S1PR1 so that the cells do not egress from the tissue. So defining a tissue resident memory cell has been challenging, and we'll use a variety of different methods to do that in our studies today. Happy to talk about that. So why do we care? As I've told you, a huge number of memory T cells are in fact these tissue resident memory cells and lodged in tissues. And in a variety, now many cases, it's clear that having tissue resident memory cells provides an advantage in protection against a range of human viral infections, as well as a very positive outlook in a whole range of malignancies. So what's clear is that if we have increased tissue resident memory cells specific for these viruses or specific for these tumors, that the outcome of both immunotherapy and overall outcome are much enhanced. And so this seems to be a worthy target in the context of simply understanding immune cell memory, but also understanding immunotherapies. And so some of the questions that we've been focused on are how are tissue resident memory populations programmed in the context of transcription factors that we know are relevant for memory T cells. And we've published a bit about this, and I'm not going to focus on that today. We also then became interested in whether or not there are tissue resident memory specific transcriptional programs. So what allows cells to leave circulation and adopt residence within a tissue? And does this inform this information? If we can learn that, how does that inform programming of other populations we would like to survive and be functional within tissues, such as tumor infiltrating lymphocytes in the context of immunotherapies for tumors? And I'll talk a bit about that, as well as we're really focused at this point on how do T cells adapt to a specific tissue. Each tissue will have its own unique context and hypoxia, glucose availability, et cetera. Each tissue is its own unique microenvironment. And so how does that influence this process? And so I just wanted to emphasize that, many studies have looked at T cells and the intrinsic factors in isolation or in a dish. And we'd really like to highlight the fact that the macroenvironment, of course, is going to be very key. There's a beautiful paper discussing specifically how obesity influences anti-tumor CD8 function in cell just this past week, but there's a whole body of literature really highlighting that a range of host metabolic and macroenvironmental influences impact the ability to make memory T cells as well as their function. And today we'll focus a bit more on the tissue context. So as I said, those unique set of signals that each tissue may have that will require distinct adaptations on the part of T cells as they enter into tissues, and where we would like them to be functional and survive for a long period of time. And this is just to highlight the fact, so this is actually incorporation of, this is a beautiful study, and I'll really just very briefly mention it. But it's an experiment showing the incorporation of 15 different nutrient tracers that allows for accounting of both circulatory and metabolic cycles and tissue TCA inputs across all of these different tissues. And what you can see is, and the point is just to see that each of these metabolic components are incorporated into cells within tissues in a very tissue-specific pattern, just really highlighting the fact that on a metabolic level, the functional activity within a tissue and the cell types within that tissue are a signature of that tissue, which means that T cells, as they leave circulation, need to take that into account as they accommodate adopting residence within that particular tissue. All right, so we started about 6 or 7 years ago now, just first asking, well, what are tissue resident memory cells? Are they just effector cells that lodged in tissue and stayed there? Are they in fact a memory lineage of T cells? And early on, Justin Milner, a phenomenal postdoc in the lab who now has his own lab at UNC, showed that very early on, so this is looking at genes that are upregulated in tissue resident memory cells compared to both circulating central memory and effector memory populations. As early as day 7, that entire tissue resident memory signature is already upregulated. So as soon as the cells hit the tissue, they adopt a unique signature of gene expression that is associated with tissue resident memory cells. And so this is just highlighted here in PCA plots, which is tissue resident memory cells in a range of tissues. Here we're showing kidney and intraepithelial lymphocytes within the small intestine are distinct from effector cells within the spleen at any given time point. And at later time points, this is nice data from Laura McKay's lab, shows that circulating and resident memory T cells have a distinct gene expression program. And then in more updated approaches, we can see here, looking at a time course of CD8 T cells responding to LCMV infection. In this branch represents the spleen versus the gut. This is as early as day 4. Cells that are the progenitors of tissue resident memory cells in tissues are already quite distinct from the T cells responding in circulation. And so this to us argued that really tissue resident memory cells were not effector cells lodged in tissues, and really highlighted the fact that they adopted a specific gene expression program associated with tissue resident memory cells. And so the first set of questions that we addressed in the lab were, well, can we identify those transcription factors that help cells adopt to residency in tissue? And we thought about it this way, which is that there are, of course, lineage defining factors and memory defining factors associated with CD8 T cells that will be true across the whole range of tissues. What makes a CD8 T cell a CD8 T cell? And that there would be a distinct set of transcriptional programs that regulated circulation versus residency. And so the first set of molecules we went after are those factors that would allow residency across the range of tissues. And so we asked for genes that were upregulated in multiple tissue resident memory populations across the range of tissues. But of course, as I highlighted at the beginning, each tissue is unique and poses a unique challenge to T cells. And we also acknowledged that there would be distinct tissue-specific modules that would allow for cells to adapt to a particular tissue. In the second half of the talk, I'll tell you about our work in that regard. But first, I wanted to highlight some published work. So Joslin took those transcription factors that were upregulated as cells differentiated, and he collaborated with Bingfei Yu in my lab, a wonderful graduate student who's now at Stanford, and Wei Wang's lab, who had adopted a bioinformatic approach, which I'm not going to go too far into right now. But it's called PageRank, and what they did was they used a range of algorithms to predict those transcription factors that were most likely to be driving changes in gene expression as tissue-resident memory cells differentiated. So using ATAC-seq and scanning for transcription factor binding sites within accessible regions linked to changes in gene expression using RNA-seq, they were able to rank those transcription factors that would be most likely to be driving changes in gene expression. And then collaborating with Matthew Pipkin and Shane Crotty's lab, we used shRNA libraries to generate targeted in vivo screens, allowing us to identify which of these transcription factors influence the ability of cells to differentiate to tissue-resident memory cells within gut, kidney, compared to spleen. And because this is published, I'm not going to spend too much time on it. Suffice it to say, two key transcription factors were highlighted in this screen. And if we knock down either RUNX3, which is interestingly enough, a lineage-defining factor for CD8 T cells, which we also found to regulate their effector function and T cell residency, and BLIMP1 were key in allowing cells to differentiate to tissue-resident memory cells within a range of tissues. Here, knocking down RUNX3, we can see that we inhibit the ability of T cells to lodge and survive within tissues. Using induced deletion studies, which are not shown here, Joslin could show that RUNX3 expression was important after tissue-resident memory cells differentiated, so they needed to continue to have RUNX3 expression to allow for survival and retention within tissues. Interestingly enough, if Joslin overexpressed RUNX3, he was able to drive more CD8 T cells into tissues and generate a larger population of tissue-resident memory cells. Now, why was this? Bingfei and Joslin took all of the data sets that were available at the time, comparing tissue-resident memory cells from a range of tissues compared to their circulating counterparts, and came up with what they called the core tissue-resident memory signature, and compared the expression of that core signature between wild-type cells and cells that were overexpressing RUNX3 cDNA. This was performed in Matthew Pipkin's lab. What we can see beautifully is that the core TRM signature was significantly upregulated when RUNX3 was overexpressed and was lost when RUNX3 was absent. Inversely, the circulating signature, the signature of genes expression that was associated with all of the circulating populations was increased dramatically when RUNX3 was not present. So we hypothesized that RUNX3 is upstream of a number of key transcription factors that regulate tissue-resident memory formation, including upstream of BLIMP1, which, as I showed you, was an important hit. And Laura McKay was able to show that BLIMP1, along with Hobbit, is important for repressing the circulating signature and allowing residency of a number of cell types within tissues. It also seems to be key in repressing KLF2, which I told you was important for preventing tissue egress, and also repression of EMS and TBET, which is also important for TRM differentiation. Now, I wanted to highlight that RUNX3 is, of course, an important transcription factor in a variety of contexts, including CD8 differentiation within the thymus and lineage commitment between CD4 and CD8 populations. And so what we were able to see is that there were a unique set of RUNX3 binding sites represented by this cloud here that it's available or accessible in CD8 TRM within the gut compared to circulation. And so really reminding ourselves that there's a tissue context-dependent targets for all of these transcription factors that allow them to have a range of functions. Okay, so at this point, what we were curious, so we'd identified RUNX3, and as I said, BLIMP1, and also NR4A1 have all been shown to be important for driving general tissue residency. And what we were curious about is, could we use this to our advantage? In fact, I'd like to remind everyone, tumors are tissues too, which we sometimes forget. And we reasoned that if cells were better adapted for survival within and functional activity within tissues, that perhaps this would provide an advantage for TIL. And so it turns out that many TIL population and tissue resident memory cells induced to viral infection have an overlapping gene expression signature. Here we can see an enrichment of these purple dots, which represent the core TRM signature. And a PCA plot highlights that there are similarities, significant similarities between TIL and TRM populations. Now, I want to emphasize that we don't think that TIL are TRM. We think that there's an overlapping signature. So there is a subset of TIL that is TRM-like. But of course, these cells have ongoing exposure to antigen, and I'm a bit loath to call them memory cells in that context. But there are genes that are turned on that allow cells to adapt to tissue residency that are also turned on in TIL. And as I mentioned earlier, there's a growing number of examples where there's a significantly positive correlation between having TIL that have the TRM signature and positive outcome in a whole range of malignancies. And perhaps this may be related to the fact that if RUNX3 is knocked out, CD8 T cells cannot accumulate within tumors. And if we overexpress RUNX3, much like we saw accumulation of TRM in the gut, we can see a significant increase, which is enumerated over here, 8 to 10-fold increase in TIL accumulating within tumors. Similarly, BLIMP1, here we're knocking down expression of BLIMP1. We can see that there similarly is a significant loss in the ability of CD8 T cells to accumulate within the tumors. So in two cases, transcription factors that we know are important for allowing cells to accumulate within tissues regulate also the ability of CD8 T cells to infiltrate tumors. We can see that overexpression of RUNX3 enhances what is already an enrichment for the core residency signature. This results in, on the bottom here, overexpression of RUNX3 results in smaller tumors and extended survival. And we think this is because not only do we have more CD8 T cells getting into the tumor, as I showed you with the flow cytometry data, but also when we look using immunofluorescence, we can see that those increased number of TIL have in fact infiltrated deeper into the tumor and have accumulated throughout the tumor, providing enhanced immune protection. This is in nice agreement with data that we reanalyzed from the Chroma lab. So Ken was nice enough to share his data with us in this beautiful method that they've developed for spatial mapping combined with single cell transcriptomics, where you can identify those TIL that were located on the outside and in the interior of the tumor, and ask what their overall gene expression is. And when Miguel did this, he could see very much as they described an enhancement of TCF and associated with the exterior or all of the TIL that were on the outside of the tumor. And we could see an enrichment of expression of RUNX3 and ID2 associated with TRM in the interior. And if you look at the overall enrichment of the CD8 tissue residency signature, we could see that it was significantly enriched in those TIL that had infiltrated deeper into the tumor and were identified as the interior. And they were in fact significantly depleted of genes associated with the circulating signature. So building the idea that having this TRM signature allows cells to accumulate deeper into the tumor and survive and provide functional activity within tissues, in this case, the tumor. And this actually translates well to CAR T cells. If RUNX3 is overexpressed in this case is a CD19 human CAR T model that Anjana Rao shared with us, we can see a significant increase in CAR T cells within these solid tumors. And in the absence of RUNX, these CAR T cells are unable to accumulate in the melanoma tumors. So building evidence that by taking advantage of our understanding of tissue residency and how cells are programmed to survive and function within tissues, we can then co-opt some of those characteristics that we would like to program T cells. So in this case, we can take the fact that TRM have heightened functional capability within tissues in spite of having inhibitory receptors. They can provide high function in the context of hypoxia. They show enhanced killing capacity. And we can borrow that and provide those programs for CD8 T cells to attack tumors. Okay, so that was one approach that we've used to kind of identify transcriptional modules that may be of utility. The second part, which I'll breeze through here, was our efforts to look at the tissue-specific modules associated with comparing and contrasting a range of tissues. So in this case, Ty Crowell, who's recently been joined by Max Hegg in my lab, have Ty compared gene expression, chromatin accessibility, and also did a single cell RNA-seq across a whole range of tissues here: spleen, fat, gut, salivary gland, kidneys, compared to spleen and blood, to ask, is it possible to identify those transcriptional programs that are unique to specific tissues? So this idea that there are unique programs to immune cells that have to infiltrate and then take up residency in tissues has been, of course, pioneered in a variety of contexts, showed beautifully several years ago now for macrophages, showing that unique transcriptional programs are turned on as monocytes infiltrate tissues and take up residence in a variety of tissues. Similar data sets exist for ILC2s and CD4 cells, and so we were interested in thinking about if CD8 T cells would interact with these transcriptional programs that had already been put forth in the context of these other cell types. So not only are CD8 T cells entering and then staying within that tissue, they are doing it in the context of a whole group of immune cells that need to provide protection within that tissue. So we know that there's a significant amount of heterogeneity. This is showing expression of the kind of classic TRM molecules CD69 and CD103, which you can see across a range of tissues are expressed quite heterogeneously. So within the gut, all of the cells are CD69 positive, or the vast majority CD103 positive, and there's a range of expression of these markers. And so we wanted to ask, how deep was this heterogeneity? We knew that there was a core signature that's upregulated, but what was going on beyond that? And in this data set, what you can see quite clearly is that we know that, as we've shown before, tissue-resident memory cells were quite distinct from circulating populations. And we could see that the gut TRM were quite distinct from the other populations of TRM that we looked at, but that each of these tissue-resident memory populations had a unique gene expression program as well. One notable distinction that we could really see is if we looked at genes associated with curated metabolic pathways, we could see clearly differences in predicted metabolic activity for first populations of cells that were circulating versus TRM. And we could see that there were certainly metabolic pathways associated with specific tissues, and I'll return to the cholesterol pathway in a couple minutes. So really summarizing a lot of data, we could see that while there are huge numbers of characteristics that are in common as cells enter tissues, there are many that are distinct. They have very tissue-specific expression of effector molecules here, showing granzyme B and granzyme A are expressed at much higher levels than TRM in the gut compared to other tissues. And this is true for expression of cytokine receptors and trafficking molecules. So clearly, and this is highlighted once again, that in using single-cell RNA-seq data, we can see that there are clear distinctions associated with cells that the same antigen specificity, the same infection that infiltrate into different tissues with a range of differences. And then we can see that there are also subsets within those tissues. There is heterogeneity as well. And that's a story for another day. I'm going to breeze through this data in the interest of time. We could also see that there were distinct chromatin accessibility differences. Here, once again, really emphasizing the difference of the TRM in the gut compared to tissue-resident memory populations in those other tissues, consistent with the fact that we could see that the gene expression programs were more similar between those other populations, and IEL was really a bit more distinct. So there are clear differences in gene expression associated with these changes in epigenetic landscape for each tissue. So going back to our approach using the PageRank method to predict transcription factors that would be important in regulating gene expression changes and differentiation of these populations, Tai asked for transcription factors that were predicted to regulate genes associated specifically with IEL, so gut TRM compared to the spleen, and was able to identify a number of factors, satisfyingly NR4A1, which I told you has already been shown to be important for TRM, and BLIMP1, which we had characterized as being important for TRM, did show up. And one transcription factor in particular that was highlighted was HIC1. Now, interestingly, that was also a very large hit when we compared IEL to other tissue-resident memory populations. So it was significantly enriched over circulating, but also enriched in the gut over other TRM populations. So this really got our attention because HIC1 was a transcriptional repressor shown by Colby-Zaff's lab to control accumulation of lymphocytes in gut, and it is retinoic acid responsive. Interestingly enough, when Tai simply asked, what are the transcription factors that all of the data set, different cell types, the data sets for these different cell types indicate are upregulated in the gut over other tissues, would there be something in common? And here the idea was really just, if all of the cells are entering into the gut, whether you're a macrophage or an ILC or CD8 TRM, you would need to make similar accommodations for that tissue. And sure enough, HIC1 was also a top hit in that comparison. So upregulated in CD8, ILCs, and macrophages as they entered into the gut. Now, Colby's lab had generated a knockout of this and had in fact reported that there was a significantly fewer numbers of CD8 T cells present in the gut when HIC1 was deleted. However, this hadn't been done in the context of antigen-specific infections. So Tai was able to knock down HIC1 and compare differentiation over time of TRM within the gut. And you could see early on there's impairment in both the gut and the kidney. And at later time points, you could see specific, really dramatic impairment in the gut compared to circulating populations, and a more moderate impairment in the kidney. Interestingly enough, when he overexpressed HIC1, he could see a significant enhancement of TRM formation in the gut TRM, but not in other tissues. And in fact, overexpression of HIC1 seemed to impair differentiation of TRM in some tissues, such as the salivary gland, really indicating that there are these transcriptional programs associated with tissue residency within specific tissues. Okay, with two more data slides, and then I'll wrap it up. I wanted to highlight, kind of circling back to what I showed you in the beginning, is that there's very distinct metabolic activity within each tissue. And Miguel Reina Campos, when he joined the lab, was really interested to understand a bit more about the metabolic activity associated with tissue residency. And using an approach much like Tai had done, he compared expression of metabolic genes across different tissues. And what he could see is that cholesterol genesis, so the genes associated with turning on the machinery that allows for a cell to make cholesterol, was uniquely upregulated in CD8 T cells within the gut. Compared to circulating cells, it was upregulated in all of the TRM, but significantly more upregulated within the gut. Consistent with this was an enhancement of cholesterol genesis by ILC2s and macrophages compared to the majority of the other tissues that were sampled. And so he hypothesized that this might be important. In addition to that, using PageRank, he was able to show that SRBF2, the gene encodes SREBP, so the transcription factor that turns on all of those genes associated with cholesterol genesis, was predicted to be important in IEL TRM. And so cells can either get their cholesterol from their diet, or they can manufacture it from glucose. And really, this requires the expression of SREBP2 or other SREBPs. In CD8 T cells, we see significant expression of the transcription factor 2 family member. And this is then essential for turning on the genes that manufacture cholesterol and is turned off by the presence of cholesterol within cells. And so Miguel wanted to ask if this was a significant enrichment. What he could see is that when he looked at T cells lodged within the gut, that they were specifically upregulated, as were all of the cells around them for SREBP2, indicating that many of the cells in that tissue microenvironment have upregulated this transcription factor. When he knocked down SREBP2, the gene that encodes, I need better terminology, he could see a specific impairment of tissue resident memory cells in the gut and a partial impairment for the salivary gland, indicating that in fact, this was an important pathway for regulating TRM accumulation and survival within the tissue. Interestingly enough, the RUNX3 knockout cells are greatly depleted for the genes associated with the cholesterol pathway, indicating that RUNX3 might actually be controlling the upregulation of this pathway as cells enter into the tissue. So I've tried to show you a range of approaches for taking cellular immunology and exploiting all of the large genome approaches that allow us to manipulate and screen for factors that regulate gene expression and cell differentiation. We've used this to identify pathways that regulate larger aspects, so the ability to leave circulation and take up residence within a tissue, but then also attempting to program cells to enter into and survive and function better within specific tissues. And I gave you two examples of that, both HIC1 and the cholesterol biosynthesis pathway. Of course, at this point, we're not quite sure why TRM have to turn on this pathway. And certainly we can hypothesize about that, and that'll be open for discussions in the future, but I wanted to highlight new data and new approaches that have exploited all of the wonderful tools we have at our disposal. So with that, I think I acknowledged most of the people who did the work as we went along. These are all possible studies due to collaborations with Shane Crowdy, Matthew Pipkin, John Chang, Jean Yo, new collaborations with Steve Benzinger, who's going to explain all things fatty acid to us, and Anjana Rao and Wei Wang. So thank you all for your time.
Thank you very much, Ananda. That was a great talk, and there are various people whose questions start with thanking you also for the great talk. So I'll join them. One of the first, actually a couple people have asked whether you know if the resident memory transcription factors like RUNX3 are important for specifically driving tissue residency by trafficking the cells there, or whether it's more about retention of the cells in the tissues.
That's a great question. So RUNX3, we don't see dramatic differences in the expression of molecules that we know are important to get cells to the gut, for instance. So our impression is that RUNX3 is important as cells hit the tissue and receive tissue-specific signals that allows RUNX3 to access a unique set of targets within that tissue. So for instance, we know RUNX3 is important for memory formation and effector function of CD8 T cells in circulation, but it becomes important for repressing Lymp1 and a variety of other signals and binding to unique sites within the tissue, perhaps because of, for instance, TGF-beta signaling as cells enter into the gut. And so those studies are underway, trying to put those in order. I think it'll be unique to each of the signals that we talked about. But Lymp1 is important, I think, in multiple places. It's important for cells to get out of the lymph node. They have to express Lymp1. So it is important in that regard for trafficking, but at the same time, we think it has unique functions once the cells hit the tissue as well. So my guess is that there'll be both kinds of signals.
Great. Actually, on a similar note, for the tissue-specific factors that you found, like HIC1, for example, are you able to, do you think, drive the tissue resident memory cell into the gut if you were to overexpress HIC1, or do you think that that's something that's sort of acquired there and more, again, for retention?
So in that case, the overexpression did allow for significantly more gut TRM to accumulate. Notably, it wasn't so much early on. It became much more dramatic over time. So there we're waiting on the RNA-seq from those studies which will allow us to say, well, what genes are upregulated and are they already upregulated in the spleen or not? We don't know. But from looking at the data, I would guess that they seem to be better adapted for survival in the gut once they're there, based on the fact that we don't see dramatically more really early on. So I think they kind of are recirculating and they get there and get some signals along with that. And similarly with the cholesterol genesis, those are just early days. We don't know, but it wouldn't surprise me if there weren't signals that primed cells or kind of got them into more likely to traffic back to a specific tissue. But we haven't seen evidence of that yet in the cases I've shown you.
Okay. I think we might have time for one more question, but there are quite a few in the Q&A. So if you feel like typing answers to those after your talk to the people who don't get their questions answered, please do that. I guess I'll do a quick one. What is the lifespan of these resident memory T cells over in the different tissues?
Well, that's a great question. We know that they can, in the lifetime of a mouse, they can certainly survive for very extended periods of time. My guess is that each tissue will have some amount of differences in turnover, but certainly within the gut, they're there for many, many months and are quite functional. In human studies with organ transplantation, it's been shown that TRM can survive many years as well. So they look to be on the lifetime of a CD8 memory cell. Early on, I think a lot of people thought, oh, well, they're lodged in tissues, they're going to be more effector-like, they're going to be more short-lived. But David Mathis' lab and Laura McKay's lab have shown these cells can proliferate and actually leave the tissue and contribute to secondary memory formation. And we've shown the same, that we can find a subset of these TRM that have more of that memory and secondary memory potential as well. So they can actually contribute to circulating memory in a secondary or tertiary response as well. So they're bona fide memory cells that stick around. Okay.
Great. Well, there are, like I said, quite a few other questions if you'd like to address those.
Thank you for all the great questions.
This.
And it's been a lot of fun. Thank you.
Yeah, thank you so much for joining us. That was a fantastic talk.